Chapter 21 is talking about the immune system. And the immune system is your body's response to foreign invaders. So when you think about a foreign invader, the word pathogen comes to mind. And a pathogen is an environmental agent that's capable of producing disease. And there are a lot of things that fall under this category, but the most common one are your microorganisms, like your bacteria, your viruses, your protozoans or parasites. Those are the ones that we're really going to be focusing on in this chapter. Okay, so there's 3 lines of defense. And the first 2 lines of defense are ones that you're born with. And because you're born with them, we call these innate defenses. The first one is the external body coverings and also the linings of your passageways in the body that have mucus-producing cells. So you have the skin and you have mucous membranes. That's your first barrier against infection. And remember, the unbroken skin is your greatest barrier to infection. The second one is if you bypass that first boundary, the skin and mucous membranes, then you have what are called antimicrobial proteins, which we'll talk about, the phagocytes, other cells that will come into play that will help inhibit the spread of infectors and prevent the disease from going any further. And out of those, all of those second lines of defense, which we'll break down and we'll talk about in more detail, inflammation is the most important mechanism because inflammation is the body's warning or SOS signal to bring in all the other elements of the immune system and let you know that there's an infection going on, there's a problem, and the body needs to mount up a defense against it. So first and second lines of defense are innate, something you're born with. Now, the third line of defense is called the adaptive defense system because it's more specific. And this line of defense is designed to attack a particular foreign substance. It takes longer to react than the first 2 lines of defense, but this one is very specific. So this is just an overview, 3 lines of defense, and then we'll go back and we'll visit each one and talk about them in more detail. So this chart gives you a breakdown. There's your innate defenses, there's your first line, your surface barrier, skin, mucous membranes, second line of defense, internal defenses, your phagocytes, your natural killer cells, inflammation, antimicrobial proteins, and fever. We will take time to talk more about these. Now, the third line of defense is adaptive, and that takes longer to develop and it's more specific. And that involves 2 populations of cells. You have lymphocytes, okay? And lymphocytes are a population of white blood cells that we talked about in chapter 17. And there's 2 populations of lymphocytes. You have your B lymphocytes. They're involved with humoral immunity. And then T cells are involved with cellular immunity. And we'll focus on this later in the chapter. So just giving you an overview of the 3 lines of defense. Now, under external barriers, this is your first line of defense. You have the skin and mucous membranes. That's in the first line. That's your major barrier. And keratin, that's found, is a protein that's deposited in your epithelial cells that make up the epidermis. That presents a formidable physical barrier to most microorganisms, and it is resistant to weak acids and bases and bacterial enzymes and toxins. So it's a very tough, resistant protein, and it makes a nice barrier being embedded and filling up those epithelial cells. The mucosa, those are your mucous membranes. This is the malt. Remember we talked about this when we were mentioning the lymphatic system. This is mucosa-associated lymphoid tissue. So it's embedded in these mucous membranes, and it helps to make a barrier in those passageways like the GI tract and the respiratory tract. Protective chemicals. These are chemicals that are in tissues that are released, especially the skin. Defensin, lysozyme, hyaluronic acid. Those particular chemical agents will help to kill off bacteria and viruses that would be on the surface of the skin. The stomach pH is around 2 to 3, and that is a very strong acid, and it can kill off just about anything that gets in there. The nasal cavity mucus, you have hairs in the nasal cavity and ciliated cells that help to trap bacteria or any inhaled particles. And then, like I said, we have the cilia. Cilia sweep the dust and bacteria-laden mucus away from the lower respiratory passageway. So a lot of protective mechanisms that are just built into the way the body's designed. Now, if you get into the second line of defense now, we have cells, okay? So if you bypass the skin and mucous membrane and that bacteria or virus gains entry past there, now you have cells to help to protect you. So we talked about these when we were mentioning the white blood cells in the blood chapter, but neutrophils, they can kill bacteria because they can take them in by endocytosis, so that's a phagocytotic process, digest them. They also can produce this toxic mixture of chemicals that if there's an area where there's a lot of bacteria, they can spew out this hydrogen peroxide and hypochlorite like a bleach solution, and it can form this killing zone that can kill off a lot more bacteria than if it was to ingest them individually. But sometimes if you have a persistent infection that neutrophils are releasing a lot of this chemical bleach, can also damage connective tissue and contribute to rheumatoid arthritis. So it's one of the things that could lead to a problem later on from persistent infections. Eosinophils, remember, they guard against parasites and allergic reactions, and they work in conjunction with the basophils that produce the histamine that cause swelling in mucous passageways. Lymphocytes, we're going to save for later because remember, those two populations of lymphocytes are involved with the specific or adaptive immune system. Those are your B cells and your T cells. And your monocytes, remember, are your wandering macrophages that infiltrate tissues and provide immunity. And examples of these cells are your Langerhans cells that you find in the epidermis, Kupffer cells in the liver, dust cells in the lungs, and microglial cells that are your phagocytes in the brain. Here's a picture that shows you a macrophage that's actively using its cytoplasmic extensions to pull bacteria in towards it and phagocytize those bacteria. Now, antimicrobial proteins. Okay, so we talked about the cells in the second line of defense. Now we're going to talk about the proteins. So the first one is called interferon. Interferon is a protein that is produced by certain populations of cells in the body, and the second antimicrobial protein is called complement. So these proteins actually attack microorganisms directly. And remember, they're not living, they're just proteins, so it's a way of kind of marking them. And they're able to hinder the microorganism's ability to reproduce. So that's generally what they do. So what does interferon do? Well, a virus, when it gains access into the body, it's not a living thing. What it does is it attaches onto a cell, it injects its DNA into the cell, and then the DNA goes and inserts itself into your cell's DNA, and then you produce more viruses. That's the way the virus works, okay? So just want to remember that that's the general way of viral infection, is making your cells into virus-making factories. And then once the virus has turned on genes to recreate itself, once that cell fills up with 2,000 to 4,000 viruses, it bursts, and those viruses go out and attack other cells. So it has to take over your cellular machinery. Okay, so the way that interferon works, virus enters a cell, and before it has access to really cause a problem, there's a gene turned on for interferon. And the cell starts to produce interferon molecules. And then the interferon binds and stimulates cells to turn on genes for antiviral proteins in nearby cells. So it goes to other cells, causes them to turn on genes to produce this antiviral protein that blocks the ability of the virus to get in the cell. So that's the idea of interferon, is it's able to protect surrounding cells from viral infection. So there is an interferon family. Gamma interferon is produced by lymphocytes. Alpha interferon is produced by other white blood cells, and they found practical uses for these interferons, that this alpha interferon is used as an antiviral drug that's used to treat hepatitis C and genital warts that is caused by herpes virus. Beta interferon is produced by fibroblasts in connective tissue, and they have found that beta interferon is used to treat the exacerbations that result in some types of MS. Complement. Complement is a group of 30 or so proteins that circulate in the blood in an inactive form. But when they're activated, these particular proteins can cause this cascade of events that results in the complement pathway. So the classical pathway is actually activated by antibodies that are coating the target cell. There's a lectin pathway that's activated by lectins on microorganisms that attack to specific sugars. And then you have the alternative pathway, which is another group of proteins. So there's a different group of proteins that start each pathway. But they all converge on this complement protein 3. And the complement protein 3, once it's activated, splits into two pieces. And you can see here that C3b causes opsonization. Now that word opsonization means that the C3b coats the pathogen surfaces, which enhances phagocytosis. So in essence, opsonization, or coating the protein with these C3bs, kind of candy coats the pathogen, so the phagocytes come and they say, yummy, let's eat that, and they take care of it, and they eat the complexes and they take them out of circulation. The C3a causes inflammation, and remember I said inflammation is the body's SOS signal, and that's going to draw in more immune cells, and those immune cells, some of them are going to release histamine, increase blood vessel permeability, attract phagocytes by chemotaxis, etc. So this is a very important process to draw in more recruits to fight off the infection. And then it continues on that if you have too many complexes that the opsonization and the phagocytosis doesn't take care of it, then the C3b is attached to the microbe, and then you see that C5 is going to split. C5b will continue this membrane attack complex that you see here, and C5a causes more inflammation. So you want to remember C3a and C5a are both involved in inflammatory responses that are going to draw in more of the immune cells and cause more inflammation. But the C5b locks into C3b, and that with C6, C7, C8, and many, many C9s, you make this membrane attack complex. I call this the Big Mac attack. And the membrane attack complex opens up a hole in the membrane so that the cell actually is lysed. And it forms a hole in the bacterial or viral cell membrane. So it forms a pore in the target cell membrane, so it can cause cytolysis. So the word lysis, remember, means to split. So far we've had phagocytosis, inflammation, membrane attack complex resulting in lysis of the pathogen. And when you summarize the whole process, there's one more thing that can happen if there's still too many complexes. So let's just back up and review. So remember that C3a and C5a result in inflammation. Phagocytosis is caused by opsonization. That's the word that is defined as the coating of the pathogen by C3b. Then you also have cytolysis that can occur if you activate the MAC complex. Remember that C3b, C5b, C6, C7, C8, and many C9s that form a ring and put a hole in the pathogen. The last thing that happens, if there's still too many complexes, that actually red blood cells can attach to C3b, and then those complexes are taken to the liver and spleen where they're stripped off and digested, and the red blood cells are returned to the circulation unharmed. So those are the four elements that are involved with the complement pathway, and depending upon the severity of the infection will determine how far this progresses. So now we're going to move into something that is still in the second line of defense. But remember, we had our cells in the second line of defense. Then we went to antimicrobial proteins, that was complement and interferon. Now the third thing is inflammation. So we want to talk about what inflammation does. It's the body's SOS signal, and it's triggered whenever body tissues are injured. It prevents the spread of damaging agents. It helps to dispose of cellular debris caused by the injury and the pathogens that try to gain access from the outside, and it sets the stage for repair, and it also alerts the adaptive immune system. That's your third line of defense. There's 4 signs of inflammation: redness, heat, swelling, and pain. Now, I understand this picture looks a little busy, but I think we want to work through it just so we see how inflammation produces an effect. And as we walk through this, I will explain everything to you, and hopefully you'll have a good understanding of what's going on when we get to the end. So the first thing is, here's your initial stimulus, tissue injury. Eventually, we want to have healing. The red boxes are the boxes that indicate the 4 cardinal signs of inflammation: heat, redness, pain, swelling. And these are the physiological responses to this initial stimulus of tissue injury. So when tissues are injured, they release inflammatory chemicals, and some of those are histamine, complement, kinins, prostaglandins, all involved with inflammation. There they are there. They look like little salt crystals being sprinkled around, and they cause a couple of things to happen. The arterioles are going to dilate, so vasodilation. Increased capillary permeability because of the dilation. This part over here, attraction of neutrophils, monocytes, and lymphocytes, I just want to leave that for a second and go through this part first. But when you have the arterioles dilating, that causes local hyperemia, increased blood flow to the area. That results, because blood's a little warmer than body temperature, so that results in an increase in the temperature, so heat, and also causes redness. And this heat causes a locally increased temperature, which also makes the cells more active, so it increases the metabolic rate of the cells that are fighting the infection, so that's a good thing. Also, increased capillary permeability, you're going to leak this very protein-rich fluid from the capillaries. It's called exudate. And that exudate fills the fluid spaces in between the cells, and because it puts pressure on nerve endings in the area, it results in pain and swelling. Sometimes, if this problem is in a joint, you may get temporary impairment of function in that joint. So this is just outlined in red because it doesn't always happen with inflammation, because not all inflammation happens in a joint. So it's a if, maybe not. But all these 4 generally will occur as signs of inflammation. Now, this increased release of exudate from the capillaries, these leak clotting proteins form interstitial clots that wall off areas to prevent the injury from going to surrounding tissues. And then you get the formation of the fibrin patch, so the scaffolding for repair is designed, so you get the clotting process going on as well. All of these eventually will result in healing. Now, attracting the neutrophils and the other immune cells. When you have a tissue injury, one of the chemicals that's released are called leukocytosis-inducing factors. These factors cause leukocytosis. Remember, leukocytosis means increased numbers of white blood cells in the bloodstream. So those leukocytes actually will migrate to the area of the injury, and then they plaster themselves up against the capillary walls. That's called margination. And then they slip in between the cells of the capillary walls through the intracellular clefts. That's called diapedesis. And then they migrate to the area where the injury is, and that's called chemotaxis, where they phagocytize the pathogen and the dead tissue cells. And the ones that take care of this are neutrophils, short-term by macrophages. Macrophages are the first ones there. Actually, if you remember this, these 3 letters, M and M. Mast cells produce the inflammation. They release the histamines. Neutrophils are drawn in. They persist, and they take care of the immediate problem. And then after they're done with their role, then the macrophages come in, and they're the cleanup men, and they clean up the area. So M, mast cells, N, neutrophils, M, macrophages. Okay, that's the order of their arrival on the scene. Then the area is cleaned of debris, and healing occurs. So there's a lot that goes on here, but this is involving the chemicals that increase the fluid and the effects of the exudate that's produced. This is the cells that are drawn in to help clear up the area, clean up any infection, and set up the stage of repair. So this is looking at that process of phagocyte mobilization again. Leukocytosis from leukocytosis factors released by injured tissue cells cause neutrophils to enter the blood from the bone marrow. Margination, the neutrophils cling to the capillary walls. You can see them here. And then diapedesis, they flatten out and squeeze through the capillary cells. And then chemotaxis, they follow the chemical trail of chemical messengers to the area where they can phagocytize the bacteria that try to gain entry through the area of the injury. So now, from there, we're going to talk about inflammatory marker that's used by the hospital to mark the presence of infection in the body. So this marker is called C-reactive protein, and it's produced by the liver in response to inflammatory molecules, and I'm just mentioning it because you might see it if you're working in the hospital and you see tests that are ordered if a person has a chronic inflammation. CRP is used as a clinical marker used to assess for the presence of an acute infection and inflammatory condition in its response to treatment. Okay, so now we move on from inflammation to fever. Fever is an abnormally high body temperature. It's a systemic response to invading microorganisms. Leukocytes and macrophages exposed to foreign substances secrete these chemicals that are called pyrogens. Pyro means fire. So these chemical messengers, these pyrogens, act on the body's thermostat in the hypothalamus, where temperature control is found, raising body temperature. So this graph shows you what's going on. There's normal body temperature, and then when the infection occurs and pyrogens are secreted, then the temperature increases, and then you reach this higher set point, higher temperature. You don't have to worry about these terms like stadium and defervescence and any of that. Just want you to get the idea of what's going on. So prostaglandins increase the temperature, and then as time goes on and the infection ends, then those chemical messengers, those pyrogens, aren't produced anymore. The population of the activated immune cells goes down, and the body temperature returns to normal. Now, I just put this little note here that aspirin and ibuprofen have their effect by inhibiting PGE, prostaglandin E synthesis. So that's how they prevent infection, or prevent fever, rather. Reye syndrome is a swelling in neural tissues of children with chickenpox or influenza, which can be triggered by using aspirin to control fever, and that's why they don't advocate giving children aspirin anymore. Usually it's just Tylenol, which doesn't have this problem. But aspirin can result in death in 30% of all victims by causing seizures and brain damage. So that's something that's very important to think about, not giving children aspirin to fight infection or fight fever. So we are done with the non-specific immune system, those first two lines of defense. And now we're going to get into more of the adaptive immune system. So that's your third line of defense. And three things you want to remember: that it's very specific. It recognizes and targets specific antigens. It is systemic. It's not restricted to the initial site. It's usually a full body response. And it does have memory. It has a stronger attack to known antigens. Now, there are two separate overlapping arms of this adaptive immune system. One is called the humoral antibody-mediated immunity, and that is put into place by your B lymphocytes. Cellular or cell-mediated immunity, that's the job of the T lymphocytes. So we have two different populations of cells that are involved. Now, when we talk about humoral immunity, and that's the immunity that comes through B lymphocytes, and those B lymphocytes are going to produce antibodies, there's a couple of ways that this immune process can occur. Now, when you have something that is active immunity means that you have memory, and that next time you have that infection introduced into the body, there are memory cells there that can knock it out before it sets up shop again. So there are two ways that you can develop natural active immunity, and the active immunity can either be naturally acquired by actually getting the infection, getting the flu, developing the antibodies, and then having protection that way, Or artificial means that you get an attenuated, which is a weakened or dead version of the pathogen that is introduced through a vaccination. And the vaccination and the exposure to those dead or attenuated pathogens allows the immune system to develop antibodies indirectly. So you're not getting the live infection, you're getting a weakened form, but the body sees it as if it was the real thing. So you develop memory, either naturally active by getting the infection or artificially active by getting a vaccination. Now, passive is a little bit different because you're not provoking the immune cells to produce the antibodies. You're getting the antibodies through injection, okay? Or you're getting the antibodies naturally through your mother. So like a little baby developing inside the mother, or getting breast milk from the mother, which contains antibodies, those are naturally acquired antibodies that the mother's producing. And they go into the baby through milk or through the placenta. But the baby's not making those or stimulating the immune cells of the baby. Anyway, babies don't have immunity until they reach about 2 years of age anyway. That's all developed early on in life. But either through the milk or through the placenta, the baby's just getting the antibodies. But once the antibodies are removed and the baby stops breastfeeding, then the antibodies go away. They degrade after about 2 to 3 weeks. Artificial is an injection, like if you get a snake bite or if you get rabies or tetanus or anything like that, then you get an immunoglobulin that's injected and you're just getting the antibodies. So in time, these will fade away. You're not stimulating a population of cells of your own to make the antibodies so you don't have memory. Remember, active you have memory, passive you do not. So naturally acquired passive immunity is from mother. Artificially acquired passive immunity comes through way of injection. So you just want to remember these 4 pathways: natural active, artificial active, natural passive, artificial passive, and how they're defined. Now, an antigen. We have to talk about what an antigen is. An antigen is a substance that can mobilize the adaptive defenses and provoke an immune response. We saw antigens on red blood cells. We had A and B. But those are our antigens. When we're talking about antigens now, we're talking about foreign antigens. And these antigens are the targets of all the adaptive immune responses. Most are large complex molecules that are not found in the body. So when we talk about our own antigens, we simply call them self-antigens, and anything that doesn't belong to us, is non-self. A complete antigen has 2 important properties. Immunogenicity. It has to have the ability to stimulate the proliferation or making a clone of specific lymphocytes. And it has to be reactive. That antigen has to have the ability to react with the activated lymphocytes and the antibodies released by the immunogenic reaction. So immunogenicity, proliferate a group of lymphocytes to respond against it, and also to react to the products of those lymphocytes. So some examples of complete antigens are foreign proteins that you find on bacteria, viruses. Sometimes they're polysaccharides, sometimes they're in the form of lipids or nucleic acids that you would find in prions or viral particles, any of those things that are considered foreign. Now, there are some antigens that are incomplete, and they have a term for that. They're called haptens. These are small molecules that are not immunogenic by themselves. Very small peptides, nucleotides, some hormones, but they may become immunogenic if they're attached to body proteins and a combination is marked foreign. And they can cause the immune system to mount a harmful attack. And not everybody responds to these, but some people do, and these are called allergies. So the immune system is not stimulated in the common way. It's like something where these particular chemicals can become attached to body tissues, and then that causes irritation. So it's an allergy. So things like poison ivy, animal dander, detergents, some cosmetics, not everybody responds, not everybody has an allergic reaction to everything, but just some people do. Now, this is an antigen. So an antigen is a huge complex, okay? So when you talk about antigenic determinants, those are little pieces or little proteins or little binding sites that are on this antigen, on this huge antigen, this huge complex. And you develop antibodies that fit like a lock and key to these antigenic determinants. Now on this picture you can see there's 3 different antigenic determinants and they have different shapes. And your body produces antibodies that are designed like a key to fit this lock, okay, and bind to these particular antigenic determinants. And some of us produce maybe this one, and then another person is genetically predisposed to produce this one. And some people can produce all 3. So it just depends on your immune system. So that's the idea of the antigen being a huge complex with all these little locks sticking out that have different shapes that will attach to specifically made antibodies produced by your lymphocytes, your B lymphocytes. Cells of the adaptive immune system. So we're going to start generally talking about the key players in this 3rd line of defense. So there's 2 types of lymphocytes, your B lymphocytes, okay? They're also called B cells. They're involved in humoral immunity. They mature in the bone marrow. T lymphocytes, T cells. These are involved with cell-mediated immunity. They mature in the thymus. So B for bone marrow, T for thymus. And antigen-presenting cells, also called APCs, they do not respond to specific antigens, but they play auxiliary roles in immunity because they actually will ingest something, digest it, put a piece of it up in their cell marker, and present it to the other players in the immune process. Okay, so T cells. They mature in the thymus under processes of negative and positive selection tests. Positive selection is that selects T cells capable of recognizing self, MHC. Now, MHC stands for Major Histocompatibility Complex Proteins. I'll break this down for you in a minute. And failures to recognize your self markers are destroyed by apoptosis. That word apoptosis means programmed cell death. So you have markers on your cells. Every cell of the body has these markers that are called MHC proteins. Your proteins are different from anybody else's unless you're an identical twin. So if you want to transplant tissue from one person to another, there has to be this MHC compatibility or the immune system will destroy those tissues, and that's called tissue cell rejection. And we see this happening a lot with people that try to get transplanted tissues. So you want to remember that the MHC marker is unique to you. Anybody else is considered foreign. Your immune system sees those tissues as being non-self because the markers are not yours. So positive selection means those immune cells, those T cells, have to recognize your markers as belonging to you. You don't want T cells that are going to go around destroying your tissue cells. So if they are, they're destroyed by apoptosis. Negative selection prompts apoptosis of T cells that bind to self-antigens, so they bind to the MHC complex, but they don't destroy it, okay? So they want to ensure self-tolerance. So you don't want cells that are going to destroy your tissue cells. You want cells that recognize your tissue cells, but recognize when those cells have a problem. So in picture form, here's what's going on. Positive selection. T cells must recognize self-major histocompatibility proteins. So there is your self, there's your cell, your body cell. There's your T cell receptor. And if it doesn't recognize, you can see there really isn't any recognition here. It doesn't fit. They've got to get rid of these before they're released into the population. So in the thymus, before they're released, if this one's not doing its job and it's not able to recognize your self markers, they're destroyed before they're released. Here you can see they recognize the self MHC, and this results in survival. Now, they recognize the self marker and they move to the next test. Now, the T cells must not recognize self-antigens, meaning that they cannot bind so tightly that they want to destroy it. So recognizing self-antigen results in apoptosis. This eliminates self-reactive T cells that could cause autoimmune diseases. So you don't want something to attach so tightly that it does destroy that particular cell. It has to be able to recognize it, identify it, and accept it as belonging to you. So if it binds too tightly to the self-antigen, it has to be able to recognize that, and there has to be a little space here so it can continue the maturation. So it has to be able to recognize that self-antigen, not become aggressive towards it, but to be able to recognize when there's something foreign that's in this pocket. Because you're going to see that these cells can ingest viruses and be able to put a little piece in here that'll be recognized by the T cell that will destroy any cell that has some type of infection, like a viral infection. So very, very important process. So as far as lymphocyte development and maturation, both B and T lymphocyte precursors originate in the red bone marrow. And then they migrate. The T lymphocytes end up in the thymus, and they develop their immunocompetency there. B lymphocytes develop their immunocompetency in the bone marrow. And then they seed the secondary lymphatic organs. So the immunocompetent but still naive, because they haven't been exposed to anything foreign yet, they leave the thymus and bone marrow. They seed the secondary lymphoid organs and circulate through the blood and lymph. And then they encounter the antigen and they become activated. And when a lymphocyte's antigen, you can see it here, there's a little Y sticking off, they bind to the antigen and the lymphocyte is activated. And in the case of the B lymphocytes, they proliferate and they differentiate. They proliferate, and that means they make many of themselves, they multiply, and then they differentiate into effector cells and memory cells. And the memory cells and effector T cells circulate continuously in the blood and lymph and throughout the secondary lymphoid organs. So there's how your populations develop from that one cell that's activated. Now, your antigen-presenting cells are the third. So you have your B lymphocytes, your T lymphocytes, and your antigen-presenting cells. They engulf pathogens. They present the fragments of the antigens to the T cells for recognition. And where you find these and what the major players are the dendritic cells that are in connective tissues and epidermis. Those two groups of tissues right there take up a large amount of the tissues in the body. Connective tissues, remember, is the most universal type or most abundant type of tissue in the body. You have macrophages that are in connective tissues and lymphoid organs, and then you have your B cells that can also become phagocytic and become antigen-presenting cells. That's the end of this part.